Hello students. So hope your NEET preparation must be going well. So many of you were writing and messaging me to make a video about this chapter, molecular basis of inheritance. So here I am with the video. So the video is a bit long because I have tried to cover each and every line of NCERT, and for each paragraph of NCERT there is one slide there. So if you are comfortable with the topic, you can increase the speed of the video, or you can pause the video, rewind it to see the important concepts. It is very important to have NCERT with you while you are going through the lecture. So let's get started with the video. We have already seen the Mendel's law of inheritance, and in that Mendel talk about factors. So Mendel has talked about factors, but he was not knowing that what is the nature of the factor. Almost 100 years after Mendel, it was identified that DNA is the genetic material. Nucleic acid. So nucleic acid actually is a polynucleotide. Nucleic acid is a polynucleotide, means it is a polymer of nucleotide. And this nucleic acid can be DNA or it can be RNA. So DNA, which is deoxyribonucleotide or deoxyribonucleic acid, and RNA is the ribonucleic acid. Here DNA mainly act as the genetic material, and RNA can also act as genetic material, but only in some viruses. Mostly RNA can function as a messenger. It can function as an adapter, as in tRNA. It also can have structural role, as in rRNA, and it has a catalytic role. If we further see the polynucleotide in detail, so polynucleotide means poly and nucleotide means many nucleotides are here. So let's see about the nucleotide. It has three composition: nitrogenous base, pentose sugar, and phosphate. Mind it, here the pentose sugar can be ribose as in case of RNA, or it can be deoxyribose as in case of DNA. And from this pentose sugar, the nitrogenous base is also connected. This nitrogenous base is connected to first carbon of pentose sugar, and this bond is called glycosidic bond. Whereas the fifth carbon of pentose sugar is connected to phosphate group, and this bond is called phosphoester linkage. This nitrogenous base present in nucleotide is of two types, purine and pyrimidine. Purine is of two types, adenine and guanine, whereas pyrimidine is of three types, cytosine, uracil, and thymine. Now, uracil is present in RNA, thymine is present in DNA. So, if phosphate is absent in nucleotide, then it is called as the nucleoside. So, nucleoside is identified by the word sine or dine, as I have told that adenosine or cytidine. So if it ends with sine or if it ends with dine, then it is nucleoside. So adenosine, guanosine, cytidine, uridine, and deoxythymidine are example of nucleoside. So nucleotide either ends with term acid or it will end with ate. Means if I say adenylic acid or adenylate, guanylic acid or guanylate, cytidinic acid or cytidinate. Two different nucleotides are connected with 3' 5' phosphodiester linkage. Means, as you can see here, the third carbon, which is also called the 3' end, has a free hydroxyl group, free OH group. Now this third carbon free OH group will connect with the 5' carbon, that is the 5' end of phosphate, which has phosphate group. So the third carbon of one nucleotide connect with the fifth carbon of another nucleotide, and here a phosphodiester bond is formed. That's why it is called 3' 5' phosphodiester linkage. As you can see here in this diagram, this is the phosphodiester linkage in which the 3' of one nucleotide is connected with 5' sugar of another nucleotide and forming this 3,5 phosphodiester linkage. Now if we see this entire one polynucleotide chain, then we see that there is a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and then if it continue, then so we can say that this backbone This backbone is made up of sugar and phosphate. Nitrogenous base projects outward. So this is how one of the end of nucleotide or polynucleotide will be 5' end, another end of polynucleotide is 3' end, and the bond is phosphodiester bond. Here two ester bond is present, 1 and 2, and here only one ester bond is present, that's why it is phosphoester bond. NCERT, one paragraph in which this important data is given and question is asked regularly from here. So remember it, that bacteriophage has 5386 nucleotide. Now why here only nucleotide is given, why not pair of nucleotide is given? Because bacteriophage Psi-174, it has single stranded DNA. Remember this important point, that they have single stranded DNA, and that is why here only nucleotide is given, base pair is not given, right? So if I ask you how many base pair is present in bacteriophage Psi-174, so you will say 0 base pair is present, because there is no pairing of nucleotide here. Then similarly bacteriophage lambda, so now you can clearly see here base pair is given, so here the DNA genetic material will be double stranded DNA, because here base pair is given. Similarly E. coli 4.6 into 10 to the power 6 base pair, and human haploid DNA is 3.3 into 10 to the power 9 base pair. So question is asked directly from here, that what is the number of nucleotide. Now if I ask you what is the number of nucleotide present in bacteriophage lambda, so base pair into 2 is the number of nucleotide. If question is asked who discovered DNA, so Miescher, and he called it nuclein. X-ray diffraction experiment was done by Wilkins and Franklin. Erwin Chargaffs has given a rule, and he said that in DNA adenine always pair with thymine and guanine always pair with cytosine. So here remember, adenine and thymine will have double 2 hydrogen bond, and guanine and cytosine will have 3 hydrogen bond. So here, so if adenine and thymine are equal and guanine and cytosine is equal, adenine plus guanine is equals to thymine plus cytosine. Adenine plus guanine divided by thymine plus cytosine is equals to 1. This is also can be said through this Chargaff's rule. So Watson and Crick has given this DNA double helical structure, and for this he has used the data given by, he has also used the X-ray diffraction data which was given by the Wilkins and Franklin. Now according to this double helical model, DNA has 2 polynucleotide chains. As you can see, this is one chain and this is another chain. So DNA has 2 polynucleotide chain. Earlier we have already seen that the backbone of this polynucleotide will be made up of sugar and phosphate. So here if we write, so it is like sugar, phosphate, sugar, phosphate, sugar, phosphate. This is the backbone. Then base pair is projected inside. Then one strand, if it is 5' to 3', another strand is 3' to 5', and this is called antiparallel. In between these two strands there is hydrogen bond, and these hydrogen bond between A and T, earlier I have said A and T is 2 bond and G and C is 3 bond. So if I give you the sequence of the nucleotide, you can easily calculate the number of hydrogen bond. Purine is double ring structure and pyrimidine is single ring structure. So this pairing between a larger structure and a smaller structure always help in maintaining the uniform distance between the strands. This is a right-handed coiling. Pitch of DNA is 3.4 nanometer. It means that one turn, let's say one 360-degree turn, it will have 3.4 nanometer. Each turn will have 10 base pair. So each base pair will be 0.34 nanometer. Now here one thing you remember, if you convert this nanometer into angstrom, so it will become 34 angstrom, and this one will become 3.4 angstrom. And this diameter, just I have explained, this one is 2 nanometer or 20 angstrom. So diameter of this DNA remains 2 nanometer or 20 angstrom. Hydrogen bond will keep these two strands together. So this is one of the way to stabilize. Another is called base stacking. So base stacking means one pair and just below is another pair, then again another pair, another pair. So this overall is called base stacking, and this will stabilize the overall structure of DNA. So Francis Crick has given the central dogma of molecular biology. Now central dogma, why it is called? Because it is common in all the living organisms. In all the living organisms, genetic information is only transferred in this way, right? Although initially it was given as a unidirectional. As you can see, information is going from DNA to RNA and from RNA to protein, but later on it was found that in some virus there is reverse transcription also. But you can say that in central dogma there is a unidirectional flow of information. So this is a central dogma in which DNA can replicate itself, called as process is called respiration. DNA can undergo transcription and it can get converted into mRNA. mRNA get translated into protein. So these all three processes comes under central dogma. Exception remain, reverse transcription is not a part of central dogma. Remember, many times students consider that reverse transcription is also a part of central dogma. No, reverse transcription is not universal. It is specific to certain virus only. Then the size of the nucleus, it is 10 to the power minus 6 meter. Now you can see the size of the nucleus is 10 to the power minus 6 meter and the DNA is 2.2 meter. So isn't DNA very much larger as compared to the nucleus? But we know that inside the nucleus DNA is there, right? So then the question arises, how can this much long DNA can be there in the nucleus, small nucleus? So the answer is DNA get condensed so that it fit inside the nucleus. So in eukaryotes, the DNA is packed with positively charged histone protein. See, this is the histone protein here and it is positively charged. Now it is histone protein, so it is made up of amino acid. Now it is positively charged because the amino acid is positively charged. It is the lysine and arginine which is responsible for the positive charge. Positively charged protein are always basic in nature. So histone is a positively charged protein. It is a basic protein. And so these amino acid, you can also say it as the basic amino acid. Now histone, it is not a single protein. It is a octamer. Octamer means it is made up of 8 subunit. H2A, H2B, H3 and H4. These are the 4 subunit and all are present 2 in number, so it becomes 8 subunit, that is octamer. Now if I say that H1 histone is a part of core histone, so answer is no. H1 is not a part of core histone. H1 is present outside the core and it is present in the connecting DNA, means this is the histone wrapped around the DNA and H1 is present outside the core histone. Now this histone is positively charged. Then how histone binds with DNA? We already seen DNA is negatively charged because of phosphate. Phosphate present provide negative charge to DNA. So phosphate provide negative charge and these lysine and arginine provide positive charge to histone. Negative and positive both will interact with each other and the negatively charged DNA will wrap around the histone octamer. Now, in this way, this entire structure formed is called nucleosome. Nucleosome consists of 200 base pair of DNA. So, nucleosome consists of a histone protein, then it is consist of DNA, it is consist of H1 histone. Nucleosome form the basic packing unit of DNA. Let's say this is one nucleosome, then another nucleosome, another nucleosome, another nucleosome, and in this way packing keep on going. In this way you can see the packing keeps on continuing like this. So can you isn't it similar like there is bead on the string? So this straight line is the string and the smaller are the beads. So that is why it is called beads on string structure. The nucleosome form beads on string structure. When seen through electron microscope, they give this appearance. Now, nucleosomes are repeating units of the chromatid. If there are total 200 base pair in the nucleus and the length of DNA is, we have already seen, 6.6 into 10 to the power 9, which is shown here. Now, and one nucleosome has 200 base pair. Then if I ask you how many nucleosomes are present in human DNA, so you will simply divide the total base divided by 200 base pair. So here you remember how many nucleosome in mammalian DNA? So 3.3 into 10 to the power 7 nucleosome. So these are the points which you have to remember. Then coming to the packaging. So till now, so if we continue with the packaging, till now what we have seen that the DNA along with histone packed into nucleosome. Nucleosome form a beads on a string structure which is called chromatin. But these chromatin fiber also further coil and condense to form chromosome. And in this is called the higher level of DNA packaging. And during this higher level of DNA packaging, additional protein is required, which include the non-histone chromosomal protein. So if question is asked in DNA packaging, so both histone and non-histone protein is required. Non-histone chromosomal protein is required. So histone protein is required for low level of packing, which is nucleosome, and non-histone chromosomal protein is required for higher level of packaging. If we talk about the type of chromatin, then you have to simply remember 3 points. First point, that euchromatin is loosely packed, whereas heterochromatin is densely packed. The second point, because it is loosely packed, it is light stained, and this heterochromatin is dark stained. Third point you have to remember, euchromatin is transcriptionally active, whereas heterochromatin is transcriptionally inactive. You just have to remember these 3 points. So first, Mendel have given the principle of inheritance. He has no idea about DNA. Then Miescher, he discovered DNA and called it nuclein. But still the molecular nature of DNA was not known. Then gradually, Sutton and Boveri has given the chromosomal basis of inheritance, and Morgan has given the concept of linkage. So this have given an idea that okay, somewhere DNA or gene and chromosome are highly linked, and these all are present in the nucleus. But still exact molecular nature we have just seen, like nucleotide, bond formation, phosphodiester bond, double helical structure, still till this point of time not exactly known. So how nature of genetic material was found out? For the first time, Griffiths has done an experiment which is called the transforming principle. So in this, what he has done, he has studied a bacteria which is Streptococcus pneumoniae. Now it causes pneumonia. Now he has taken two strains of the bacteria. One is called the S strain or smooth strain, which is virulent. Virulent means which can cause disease. Now it has a polysaccharide code. Now tell me, what is this polysaccharide code called? So the capsule, right? Capsule is, in cell chapter we have seen that bacteria has an additional layer of polysaccharide code called capsule, which make the bacteria virulent, and that capsule is absent in our strain, so it is non-virulent, it is rough, and it has no code. Now very simple, S strain, it is cause disease, so live S strain will kill mice. R strain which do not cause the disease, so live R strain, the mice will easily survive. Now if we heat kill this S strain which cause disease, but now we are heat killing it, so if we heat kill, then still the mice survived. Then if we heat killed S strain, means we are now mixing heat killed S strain with live R strain. Now live R strain, mice survived, and heat killed S strain, mice survived. So in both the case separately mice survived, but now when they both are mixed together, surprisingly the mice died, right? So in these two separate conditions the mice survived, but why it got dead in when they both are mixed together? So Griffith has, from this dead mice, he isolated. So when he isolated, he find live S strain, right? He put heat killed S strain and he isolated live S strain. So it was very strange. A cell which is dead cannot be alive, right? This is against the principle of biology. So what is the only possibility? That this live S strain get transformed into live R strain get transformed into live S strain. So this is the only possible explanation. So he has concluded that the R strain bacteria get transformed into S strain, and this is due to the transforming principle. Means something from R strain has entered, something from heat killed S strain has entered into the R strain, and this has transformed the R strain into S strain, and this something he called it as the transforming principle. But he could not explain the genetic material of this transforming principle. He didn't say that this transforming principle is protein, or it is DNA, or it is RNA. Griffith could not explain the biochemical nature, but then three scientists, Avery, McLeod, and McCarthy, they have performed some experiment, and they have concluded that the biochemical nature of transforming principle was DNA. So this is the climax information. How, let's see step by step how they have proceeded with the experiment. So they have, so only there are three cases. It can be protein, DNA, or RNA. So what he has done, he has taken the heat killed S strain. Now you know that the transforming principle is coming from heat killed S strain. So he has taken the heat killed S strain, and from that heat killed S strain he isolated protein, he isolated DNA, and he isolated RNA, because these three can be the transforming principle. Then he treated the R strain with protein, DNA, and RNA one by one. See, the purpose is that this R strain is getting transformed into the S strain, because of these transforming principle. First, he has taken protein or protease, which will digest the protein, right? So when protein was digested, there was no effect on transformation, right? Means even if you destroy protein, transformation still happen. Means protein is not the transforming principle. RNase was used, means which will digest RNA, still there was no effect. But when DNA was digested using DNase, then the transformation stopped. So this given a conclusion that the DNA is the transforming principle.
So Hershey and Chase, he has given the definitive proof that DNA is the genetic material. So here he has used a bacteriophage. Bacteriophage is a virus which infect bacteria. Actually, as we can see here, bacteriophage can infect bacteria and it will inject its genetic material inside the bacteria, and it will force the bacteria to produce the new virus. Now, the genetic material which enter inside the bacteria, at the time of Hershey and Chase, it was not known. So the question was whether it is a DNA or it is a protein. Now, before that, let's first understand the structure of this bacteriophage. So here if we see, this bacteriophage has two parts. One is the outermost coating, which is protein, and the innermost part is the DNA. Now, the fact is that the DNA has phosphorus, which is not present in protein, and protein has sulfur, which is not present in DNA. So in order to radio label DNA, radioactive phosphorus was used, and this radioactive phosphorus was P32. Why it was used? Because phosphorus is only present in DNA. So with P32 we can easily radio label DNA, and with the help of S35 we can easily radio label the protein. This experiment has three steps. First step, which is the infection, in which the bacteriophage infects the bacteria. Here, bacteria will inject its genetic material inside the E. coli. In the second step, the attached virus on the surface of E. coli is blended, which is the blending step, and it is separated from the E. coli. In the third step, which is the centrifugation, different components are rotated at a very high speed and they are separated on the basis of their mass. After centrifugation, the heavier objects settle at the bottom called pellet, and the lighter object remain in the supernatant. Now, here if we see the result of this experiment, so when the protein was radio-labeled, in that case the E. coli was non-radioactive whereas the supernatant was radioactive. Whereas when the DNA got radio-labeled, then the E. coli is radioactive and the supernatant is non-radioactive. So this gives us an idea that the protein is not entering inside the cell, whereas DNA is entering inside the cell. And hence we conclude that DNA is the genetic material. Note here that not always DNA is the genetic material. RNA can act as genetic material in some of the virus. An example are tobacco mosaic virus and QB virus given in NCERT. Now, in order to be called as the genetic material, every molecule must have certain common character. And these character as listed here are a genetic material must be able to replicate. It should be chemically and structurally stable. It allows slow mutation and it should express Mendelian character. RNA world hypothesis says that RNA was the first genetic material. Now, what are the evidences? So RNA can store genetic information. RNA at the same time can also act as catalyst, and this is essential for several life processes: metabolism, translation, splicing, and these all life processes revolve around RNA. So it is very logical to say that RNA in the early phase were performing all these functions, and along with that they were also acting as the genetic material. But RNA is unstable because of presence of ribose sugar, so DNA gradually evolved and it replaced RNA as the genetic material due to better stability. If we compare DNA and RNA, then we can simply say that DNA and RNA both can replicate because both can show base pairing. DNA chemically more stable than RNA because DNA lack a hydroxyl group present on the 2' carbon, which prevents it from degradation, and there is thymine present in DNA instead of uracil, which provide an additional stability. RNA being unstable mutate faster than DNA, and this lead to faster evolution of RNA virus. With respect to expression of trait, RNA can directly express and synthesize protein, whereas DNA cannot directly synthesize protein. It depends on RNA for protein synthesis. So seeing all these points, we can conclude that DNA is the preferred genetic material because of its stability, whereas RNA is better for transmitting the genetic information. Now let's see DNA replication. Watson and Crick were the first to give the double helical model, and he also suggested the semi-conservative replication process for DNA. Now he said that each template will synthesize a new complementary strand. Means each newly synthesized DNA has one parental strand and one newly synthesized strand, and that is why it is called semi-conservative replication. Watson and Crick has given a theoretical explanation of semi-conservative replication. The experimental proof that DNA is the genetic material was given by Michelson and Stahl. Michelson and Stahl has proved that DNA is the genetic material using a very important experiment, and in this experiment he used N15 heavy isotope. N15 heavy isotope is not a radioactive isotope, mind it. It is a heavy isotope, means its mass is high, and because of presence of N15 the DNA become heavy. So N15 will be heavier and N14, which is the normal nitrogen, will be lighter. So in this experiment DNA was analyzed using CSCl density gradient centrifugation. Now here if we see this experiment in little detail, then the original first DNA which was synthesized has N15 nitrogen. So both the strand are N15 here. So both strand are N15, then this N15 will be heavier. Now when it is N14 and N15, it will be called as the hybrid. And when N14 and N14 is there, then it is called light chain. So as you can see here in this it starts with both strand N15. Then in first generation, that is after 20 minutes, you get 2 DNA. So one DNA become 2 DNA, and here both the DNA are hybrid, means one strand is N15, another strand is N14. So it has a hybrid strand of N14, N15. Then in the second generation, that is after 40 minutes, these two DNA become 4 DNA. You can see these two DNA become 4 DNA, and here the two hybrid DNA is here, and then two light chain DNA is here, because the N14 will synthesize a new N14 strand, and N15 will also synthesize a N14 strand. Now if we continue it to the third generation, then in third generation 4 DNA will become 8 DNA, and here out of these 8, 2 will be hybrid and 6 will be the light chain. So one type of question is asked about the number of DNA. So in this kind of question, they will ask you that after some generation, how many number of DNA. So let's say after first generation, the number of DNA will be 2. After second generation, the number of DNA will be 4, and so on. Third generation, it will be 8. So the general formula for number of DNA after generation is 2 to the power n, where n is the number of generation. So you can easily now find out the number of DNA after a given generation. If I ask you, let's say after third generation, tell me what will be the ratio of hybrid to light chain. What is the ratio of hybrid to light chain? So such question how we can see. Now here, as we know that after third generation, the number of DNA will be 2 to the power n, that is 8. Now out of this 8, in any generation, always you will get 2 hybrid. So 2 hybrid. So how many will be the light chain? 6 light chain. So what will be the ratio? So the ratio of hybrid to light chain will be 2:6, that is 1:3. After fifth generation, tell me the ratio of hybrid DNA to light DNA. So here after fifth generation, 2 to the power 5, that is 32 DNA. After fifth generation, 32 DNA. Out of that, how many will be hybrid? So in every generation there is only 2 hybrid, and how many will be the light chain? So 30 will be light chain. So what will be the ratio between hybrid and light chain? So hybrid is 2, light chain is 30. So the ratio will be 1:15. Now let's see DNA replication. So DNA replication require an enzyme, and this enzyme is called DNA dependent polymerase, or simply DNA polymerase. It is called DNA dependent because it uses a DNA template to synthesize a new DNA. Now this DNA polymerase is highly efficient. It is highly efficient because it can replicate 4 to the power 6 into 10 to the power 6 base pair in just 18 minutes. Means it has a replication rate of 2000 base pair per second. So this is very high rate of replication. Not only rate is high, but the accuracy is also very high. And if there is any error in the replication, then it will lead to the mutation. Now this DNA replication is a energy intensive process, and here the deoxyribonucleoside triphosphate provide the energy. This nucleoside triphosphate is basically a nucleotide in which there is this pentose sugar which has a nitrogenous base attached, and here instead of 1 phosphate they have 3 phosphate, and the energy is present here in the phosphate group. So DNA replication take place with the formation of a replication fork. Now this DNA not separated, or it do not entirely unwind. Only, for example, let's say if this is the DNA, then it's not that the entire DNA get separated. It is only a part of DNA which will separate, and the replication fork is formed here. So as you can see here, the replication fork is formed. Now why it is a small opening only? Why not the entire DNA is separated? Because it requires very high energy, which is not feasible for the cell. So this small opening is called the replication fork, and it is the site where replication will occur. It is also the site where DNA polymerase activity take place. Now both the strand of DNA act as a template, and one of the template is called 3' to 5' template, another template is called 5' to 3' template. In the 3' to 5' template, replication occur continuous, and that is why it is called leading strand. Whereas in 5' to 3' template, the replication is discontinuous, so it is called lagging strand. So there is a leading strand in which the continuous synthesis of DNA take place, and there is a lagging strand in which discontinuous synthesis of DNA take place. When the DNA is synthesized in a discontinuous manner, then it is synthesized in small fragments, and each of these fragments are called Okazaki fragments. So these are the Okazaki fragments. As you can see, these Okazaki fragments will ultimately join together and it will become a single fragment. Here, this joining of Okazaki fragment is done with the help of an enzyme, DNA ligase. So DNA polymerase synthesize the DNA in 5' to 3' direction, whereas DNA ligase, it joins the Okazaki fragment. The DNA replication always begin at a specific DNA sequence, which is called the origin of replication. No DNA can replicate without origin of replication. Vector is used as a in recombinant DNA technology as a plasmid because vector has origin of replication. So vector has origin of replication, and the main DNA of prokaryote or eukaryote also have the origin of replication. There is a coordination between DNA replication and cell division. As you can recall that during cell division, the nuclear division is always coupled with the DNA replication. Replication take place in S phase, and then the karyokinesis take place. In mitosis, one replication, one karyokinesis. In case of meiosis, one replication and two karyokinesis. Failure of cell division after DNA replication can lead to polyploidity. Transcription, that is the formation of RNA from DNA, also follow the base pairing rule. Here A, adenine, will pair with U because we know that in RNA there is no thymine. So A instead of thymine will now pair with uracil. Main difference between replication and transcription is that replication, the entire DNA is copied, but in transcription only a segment of DNA is copied. Also, in case of DNA, both the strand act as the template, whereas in this case only one strand will act as the template. Reason? Because if both the strand act as the template, or there is a transcription of both the strand, then there will be a difference in the code of RNA, which will lead to formation of different protein, and this will create a confusion in gene expression. Another reason is that if both the strands synthesize RNA, then these RNA will be complementary to each other. And if they are complementary to each other, then they will form a double-stranded RNA. And this formation of double-stranded RNA will prevent the protein synthesis. Transcription takes place in small units called transcriptional unit, which has mainly three parts: promoter, structural gene, and terminator. The promoter, as you can see here, it is the region where the RNA polymerase will come and bind. Whereas the structural gene, it is the place where the sequence is transcribed into RNA, whereas the terminator, it will signal the end of the transcription. The transcriptional unit has two strands, template strand and the coding strand. As you can see, the template strand is from 3' to 5', whereas the coding strand is from 5' to 3'. So template and coding strand both are the strand of DNA, so they are anti-parallel. Template strand act as the template for the RNA synthesis. So the RNA will be complementary to the template strand, whereas the coding strand, the RNA synthesized will have a sequence similar to the coding strand. If you compare the template strand and the RNA sequence, so here you will observe that the template strand and the RNA sequence, they both are anti-parallel to each other. See, one is 3' to 5' and the RNA sequence is 5' to 3'. So this is one way to simply find the orientation or the how is the direction of this two strand. Then Coding strand, if you see, coding strand has the polarity 5 prime, 3 prime, and the RNA sequence also has the same polarity. So the polarity of coding strand and the RNA sequence is same, whereas the polarity of template strand and RNA sequence is opposite. So this is one way to identify. Then second factor you need to observe that the sequence of template strand and the sequence of RNA both are complementary to each other. So let's see here. Here you can see there is A, here U, then T, here is A, here is G, here is C, C, G, A, U. Means what? It is complementary to each other. In case of coding strand, you will not see any complementarity. It is exactly same as the coding strand. The only difference is instead of T, it will have U. Rest all the sequence will be the same. Another important point to observe here is the position of promoter and position of terminator. So one convention is that promoter and terminator position always is considered with respect to coding strand. So as you can see here, coding strand is given, which is 5 prime to 3 prime. So if question is asked, which side of the coding strand is promoter present? So it is present in the 5 prime end of the coding strand, and terminator is present 3 prime end of the coding strand. The side where promoter is present, this side is called the upstream. This is called upstream. And this side is called the downstream. There is upstream and there is downstream. Now, remember, side of promoter is upstream, side of terminator is downstream. Now, the same question if I ask that with respect to template strand. So, with respect to template strand, the promoter is present toward the 3 prime end of the template strand and the terminator is present toward the 5 prime end of the template strand. So in this way, from here statement based question can be made regarding the position of promoter, regarding the position of terminator, find out the RNA sequence, find out the polarity of template strand, coding strand, RNA sequence. Promoter basically determine the template strand. Switching promoter and terminator can reverse the strand rule. For example, let's say now here this promoter, if I change the promoter to terminator and if I shift the terminator to promoter, if promoter goes to terminator, terminator goes to promoter, then the coding strand will become template strand and the template strand will become coding strand. Now let's see this with the help of a question. So here if let's assume that the template strand is 3 prime A T G C A T. Let's say this is the template strand. And now the question is find out the RNA sequence and the coding strand. So let's first find out the RNA sequence. So RNA sequence. So we know that the RNA sequence will be anti-parallel to the template strand. So here template strand is 3 prime. So here it should be 5 prime. Then RNA sequence is complementary to the template strand. So A will be, instead of T there will be U. T A G C C G A U and T A. So this will be the and 3 prime. So this will be the RNA sequence here. Now if I ask you what is the coding sequence, so coding sequence is exactly complementary to the template. So coding strand will be 5 prime and it will, in a sense, it is also same as the RNA sequence except the sequence of U. So 5 prime T A C G T A and 3 prime. So in this way, question can be asked about, you may be given a coding strand, then you are asked RNA sequence. You may be given an RNA sequence, asked about template strand. You may be given coding strand, asked about the RNA sequence. So any one of it can be given and other can be asked. Gene is the functional unit of inheritance and it will code for RNA or protein. Remember, gene can code for RNA also and it can code for protein also. See, all the gene produce RNA and then RNA produce protein, but not all RNA will produce protein. See, one thing. RNA can be mRNA, RNA can be rRNA and RNA can be tRNA. Now out of these three, mRNA will form protein, but not these two will form protein, but these all two will also be formed from the gene. So gene will code for both RNA and protein, whereas a cistron will only code for protein or polypeptide. So this is a very important difference between gene and cistron, that gene can code for RNA as well as protein, whereas a cistron code for a single polypeptide. Cistron can be divided into monocistronic or polycistronic. Monocystronic means it will code for one protein as it is found in eukaryote, and polycistronic which code for multiple protein which is found in prokaryote. In eukaryote there is a concept of split gene which is found in eukaryote, because in eukaryote out of all the sequence there can be exon and intron. Exon is called the coding sequence, whereas the intron is called non-coding sequence. Coding sequence will be present in matured RNA, whereas the non-coding sequence, it is removed during the processing of RNA. Apart from this, there are several other regulatory sequences present. These regulatory sequences can control the gene expression. These regulatory sequences can be promoter or regulatory elements. If we see the type of RNA, there are mainly three types of RNA: mRNA, tRNA and rRNA. mRNA is the messenger which act as the template for protein synthesis, whereas tRNA, it brings amino acid and it deciphers the genetic code, means as per the genetic code it will bring the amino acid. It is also called as the adapter molecule, whereas rRNA, it has a structural and catalytic role. It is a component of ribosome. Remember, it's the rRNA which can act as catalyst and it is called ribozyme. Transcription involves three steps: initiation, elongation and termination. During the initiation step, the RNA polymerase will bind to the promoter. When it binds to the promoter, here there is a sigma factor is present, and this sigma factor will help in the initiation of transcription. At this point, the DNA unwind and the transcription will begin. So in the beginning or in the initiation, sigma factor has an important role. Once the initiation is done, then the sigma factor will be released, and now the RNA polymerase will move onto the template strand in 3' to 5' direction, and this will synthesize the RNA in 5' to 3' direction. Remember, the RNA synthesis takes place in 5' to 3' direction. The DNA synthesis was also taking place in the 5' to 3' direction, and here also the nucleoside triphosphate is serving as substrate. So it also serves as substrate and it also serves as the source of energy. So NTPs serve two purpose: it acts as substrate and also help in providing energy to the process. Then the last step, termination. During elongation, the RNA will keep on elongating. Then finally, when the terminator sequence come, then the rho factor will come and attach to the RNA polymerase, and this rho factor will stop transcription, and in this case the RNA is released. So in these three steps, the RNA is synthesized and it is released. In eukaryote, the transcription involves three types of RNA polymerase. These three types of RNA polymerase are RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Here, the RNA polymerase 1 synthesize rRNA, and specifically 28S, 18S and 5.8S rRNA. You must be knowing that the rRNA is of several types. It can be of larger subunit and smaller subunit. Here see, 5S rRNA is not a part of here. 5S rRNA will be formed by RNA polymerase 2. So RNA polymerase 1 synthesized rRNA except 5S rRNA, whereas RNA polymerase 2, it is the precursor for mRNA. It will form the mRNA, whereas RNA polymerase 3, it will form tRNA and 5S rRNA and small nuclear rRNA. So function of all three RNA polymerase, keep it in mind. Then once the precursor of mRNA, that is hnRNA, which is when it is synthesized, then it undergo processing, and the processing of hnRNA involve splicing, which is the removal of intron and joining the exon. Remember, intron is the non-coding part, whereas the exon is the coding part. So intron is removed and exon is joined. Another modification is the capping. Here, methyl guanosine triphosphate is capped at the 5' end. It is also called as 5' capping. Then in the tailing part, 200 to 300 adenylate residue is added at the 3' end. After these three steps, now this mRNA is formed, And this newly formed mRNA, which is formed in nucleus, now it will be exported to the cytoplasm for translation. Till now everything has taken place in the nucleus. Now this mRNA will be transported into the cytoplasm for translation. On the other hand, this is about eukaryote. In prokaryote, transcription and translation occur simultaneously, because in prokaryote there is no need of processing of hnRNA. George Gamow for the first time proposed that genetic code is triplet in nature, that is, each codon has 3 nucleotides. But George Gamow hasn't performed any experiment to prove that genetic code is triplet. The experiment performed was by subsequent scientists. So for the first time, Hargobind Khurana, he developed a chemical method for the synthesis of RNA, and here he said that genetic code is triplet. For that he has used homopolymer and copolymer. So the key word here is he used chemical method and he formed homopolymer and copolymer. Then another scientist is Marshall Nirenberg. He used cell-free system for protein synthesis, and this lead to deciphering of the genetic code. Another scientist, Severo Ochoa, so he discovered the enzyme polynucleotide phosphorylase, and this enzyme polynucleotide phosphorylase help in RNA polymerization without a template. So till now what we understand that for the formation of RNA you need a template, but if you want a template independent RNA synthesis, then this enzyme will be helpful. Now here you can keep this in mind that he basically explained the enzymatic synthesis of RNA. Then following that, a genetic code checkerboard was given, and this genetic code checkerboard, basically it is a systematic approach to determine the triplet code sequence for the amino acid. So summarizing all the characteristic of genetic code. So genetic code is triplet in nature, that is each codon has 3 nucleotide. Each codon has 3 nucleotide and it code for 1 amino acid. Genetic code is degenerate, means some amino acid are coded by multiple codon. Genetic code is continuous, it means that there is no punctuation. We can also call it commaless. Genetic code is universal, means the same code is used by all the organism, but here there are certain exceptions also. In very few cases the genetic code variation is found. AUG is the start codon which code for amino acid methionine. It is also called the initiation codon. Every mRNA, the coding sequence always start with AUG. The stop codon include UAA, UAG and UGA. This stop codon do not code for any amino acid, but they help in termination of the translation, so they are called termination codon. Further, if we see this RNA sequence, so which is starting with AUG, and then there are several triplet codon present over here. So if we see this checkerboard, so here you can clearly see that AUG coding for methionine, and then following that all are amino acid coding phenylalanine, but the code are different. As you can see, somewhere it is UUU, somewhere it is UUC. So here this is the degeneracy of codon, and due to degeneracy there is ambiguity because multi-codon can code for same amino acid. So this is what degeneracy is. Here in this checkerboard you can see there are several codon present over here, 3-letter codon, and this codon code for a particular amino acid. Now here you don't need to remember all the codon, but yes, you need to remember in detail about all the codon given here in the text. For example, UUC code for phenylalanine, AUG code for methionine. In the same way, if some other is also given in the text, then that is important from exam point of view. Further, if we see, this genetic code can undergo mutation, and this mutation can be point mutation, means change in a single base pair, or this mutation can be frame shift mutation, which include insertion and deletion of sequence. This point mutation example is sickle cell anemia, where you see that in the GAG codon, adenine is replaced by uracil and GAG become GUG, which change the amino acid glutamate to valine, and due to that there is a change in beta globin gene which cause sickle cell anemia. In frame shift mutation, there is insertion and deletion. And insertion and deletion of one or two base shift the reading frame and alter the protein. Whereas insertion or deletion of three base pair or its multiple do not shift the reading frame. This is an example where three letter word is used and it is representing a reading frame like Ram has red cap. So this is a reading frame. Every word has certain meaning. But now let's say you are inserting a letter B and this B letter you are inserting here just before red. So now you can see first two letter has the meaning Ram has, but after that you can see the meaning has changed, so there is a frame shift mutation. The same will happen if you insert two base pair. Same reading frame will shift. But what if you insert three base pairs? So this is the example, big. So if you insert three, then only one codon is added. Rest of them are having the same meaning. For example, still you can see Ram has, just this word is added, big, but red and cap still you can have the meaning. But here you can see, you can find that red and cap is gone. But here still red and cap is there. Means there is no frame shift mutation if three or multiple of three bases are added into a sequence. Now, moving on to translation. So we have already seen that mRNA is formed after transcription. During the translation, this mRNA will be converted into protein. So now let's see this process. So in this process, the first step which occurs is charging or amino acylation of tRNA. Here, amino acid get attached to a specific tRNA. So as you can see, this is the tRNA which is uncharged. Now here you can see one amino acid is added along with ATP. So this is an active process where on one end of tRNA and which end? The 3' end. At the 3' end, the amino acid get attached and this is called charging of tRNA or amino acylation of tRNA. This process require ATP. So this is an active process. In this process, you can see an enzyme is also involved and that enzyme is called amino acyl tRNA synthase. Francis Crick proposed tRNA as the adapter molecule. Now if we see the structure of tRNA, tRNA has anticodon loop and it also has an amino acid acceptor site. The 3' end site is amino acid acceptor site, whereas this anticodon loop bind to the codon which is present on mRNA. Each tRNA here, each tRNA is specific to one amino acid. So for each amino acid, there will be a separate tRNA. There is a special type of tRNA called initiator tRNA which recognize the AUG, which is the start codon. So if let's say AUG is the start codon, so the initiator tRNA will have anticodon and that anticodon should be UAC, and here it will also have an amino acid on the 3' end and this amino acid here is methionine because the AUG code for methionine. So this is the codon, this is the anticodon, and this part is the amino acid. So here remember we have already seen that the tRNA present here is already charged in the previous step. Initiation is the first step of translation, and here first ribosome bind to mRNA at the AUG site, that is the start codon, and on this start codon there will come a initiator tRNA as we have seen previously, and that initiator tRNA will have a anticodon that is UAC and that will have a amino acid which is methionine. So can we say that every time the first amino acid will be methionine because the start codon is always AUG and AUG always code for methionine? So this is the initiation step. Once this initiation is done, then the ribosome will move along the mRNA in 5' to 3' direction. It will move and then the next codon will be exposed and this codon will again be bound by the another tRNA which is specific to that codon, and then the amino acid from one tRNA get transferred to another tRNA like this, and here a peptide bond is formed. So let's say this is one amino acid, one tRNA with amino acid. Let's consider this is AUG. Then the next amino acid is GUG. So here next tRNA will come. It will also have amino acid, and here the anticodon will be CAC. Now once these both are attached, then this amino acid will get transferred onto another amino acid, forming a peptide bond. So in the elongation step, peptide bond is formed between the amino acid. And in the formation of this peptide bond, there is a very important role of rRNA, which act as ribozyme. Following that, the ribosome move codon by codon along the mRNA in 5' to 3' direction, and it will lead to a continuous addition of amino acid and subsequent formation of peptide bond. Once a amino acid is transferred from the tRNA, then this tRNA again become discharged and it is released from the ribosome. So basically, the uncharged tRNA is going out from the ribosome and the charged tRNA is coming in the ribosome. And in this way, the entire process of translation is continuing during the elongation. Now, as the stop codon, that is UAA, UAG and UGA is reached, at that point, a release factor bind to the ribosome. Now here you have to understand that UAA, UAG and UGA do not code for any amino acid. There is no specific tRNA for these codon. So instead of tRNA, release factor will come and bind at the site, and this release factor will release the polypeptide, and it will also lead to the disassembly of the ribosome. So this will conclude the translation, and the polypeptide which is formed will be released into the cytoplasm. So here some key components of translation, let's see. So one of the key component of translation is ribosome. So that is why it is also called as the cellular factory for protein synthesis. Another important factor for translation, as I said earlier, is the rRNA, and specifically 23S rRNA in bacteria, which function as ribozyme. So this is the catalyst for peptide bond formation. Apart from this, it also have, the mRNA also have untranslated region, which is actually do not have any codon, and it is not translated, and it is present at the 5' and 3' end, but these are very important for efficient translation. So once again, if I repeat, untranslated region are not having any codon, they do not code for any amino acid, they are found at the 5' and 3' end, and they are very important for efficient translation. Now the polypeptide formed undergo further folding and modification to become functional protein. As we have seen in biomolecule chapter, that they first form primary structure, then secondary, then tertiary structure. So in this way, we have completed the central dogma of molecular biology, in which we have seen how RNA is formed from DNA in the process of transcription, and then we have seen how RNA. So this overall formation of protein from DNA is the expression of a particular gene. So this gene expression can be regulated at several levels. So gene expression regulation refers to, it is controlling the protein synthesis at different stages. So basically all the protein are not synthesized every time. It depends on age, time, condition, environment. So something is regulating the expression of these protein, and that we will see under the gene expression regulation. So this regulation can occur at which level? So it can occur at transcription level, it can occur at processing level, it can occur at transcriptional level, where the formation of primary transcript, that is mRNA or hnRNA is basically stopped. At the processing level means during the hnRNA to mRNA formation, it is regulated, or mRNA which is formed, it is not transported from nucleus to cytoplasm. It can also be done at the translational level, that is, it controls the protein synthesis by inhibiting certain enzyme which is helpful in protein synthesis. So what is the need of gene regulation, or why a gene is regulated? So every gene is expressed to perform a specific function, and specific function is performed only in the specific condition. So let's say E. coli. So E. coli produce beta galactosidase enzyme to break down lactose into glucose and galactose. Now the purpose is to break down lactose. So beta galactosidase enzyme is required only if lactose is present. So beta galactosidase enzyme is produced only in the presence of lactose, and it is absent in the absence of lactose. There are certain environmental and metabolic condition also which can regulate the gene. In certain metabolic condition, for example, during digestion you need certain enzyme in more quantity. In certain environmental condition, a certain types of enzyme hormone is released in the body. Similarly, when there is embryonic development, so there also it require a coordinated gene regulation. So you can see that gene regulation is required at different stage of life, different condition of or different time, and on the basis of environment. Gene regulation in prokaryote is mainly done at the transcriptional level. In this case, the RNA polymerase activity is controlled by the regulatory protein. So there can be activators which increase transcription. There can be repressors which decrease transcription. On the same side, there will be operator which will bind where the repressor will bind. So each operon has a specific operator and repressor. This lac operon was discovered by Jacob and Monod. Lac operon regulate the lactose metabolism in E. coli. So here there are certain regulatory gene, that is I gene, which is present here. This I gene code, it forms a repressor protein. As we can see here, this is the I gene and it code for a repressor protein. Apart from this, it also has structural gene. The structural gene are present as Z, Y and A. So Z gene produces beta galactosidase which break lactose into glucose and galactose. Y gene produces permease which helps the lactose to enter into the cell. A gene, it produces transacetylase, but its function is less understood. Apart from lac operon, there are many other types of operon like tryptophan operon, arginine operon, histidine operon, valine operon. So we will study only lac operon in detail. So now here this is a diagrammatic representation of how Z gene is coding for enzyme beta galactosidase, Y for permease and A for transacetylase. In lac operon, lactose act as the inducer which switch on the operon. A low basal expression of lac operon is always present, which allow some lactose to enter inside. If there is no lactose in the cell, then in this I gene which form repressor, the repressor bind to the operator. When the repressor bind to the operator, there will be no transcription. If there is no transcription, then there will be no formation of protein. So no lactose, repressor will bind to the operator and this will block the RNA polymerase and this will lead to no transcription. But if lactose is present, then it will bind to repressor and repressor get inactivated. As you can see, if lactose here is present, that is, you can see the inducer is present here. So it is binding to the, as you can see here, it is binding to the repressor, and as it bind to the repressor is inactivated. So now you can see repressor is no longer going and binding to the operator. And if it do not bind to the repressor, then the RNA polymerase transcribe the gene and all the polypeptide beta galactosidase, permease and transacetylase is formed. Only lactose can act as inducer. Glucose and galactose cannot act as the inducer. And expression of this lac operon lasts as long as the lactose is present. This lac operon, with the help of lactose, it is under negative regulation. But lac operon also show a positive regulation. Human Genome Project. The purpose of this project was to find the DNA sequence or the genetic makeup of an organism, that is the human in this case. Now, different individual will have different DNA sequence. So basic aim of this project was to decode the complete human DNA sequence. It started in 1990, and this cost estimate of this project is about $3 per base pair. So there will be a $9 billion expense for this project. If printed in the form of book, it will require 3,300 books of 1000 page each. Now this has generated a massive data, so it required high speed computing, and this lead to emergence of field of bioinformatics. The main aim of this human genome project was to identify 20 to 25,000 genes present in human DNA, to determine the sequence of 3 million base pairs, store these information in a database, and to analyze this data to be used as a tool. Now, these data selectively to be transferred to industries for better use, and also to address the ethical, legal and social issues. The project, which started in 1990 and completed in 2003, so it lasted for 13 years. It was led by US Department of Energy and National Institute of Health in collaboration with different countries. Here, if we understand the DNA variation, it can help us to diagnose, treat and prevent several diseases. Later on, non-human organisms were also sequenced for research, and it included bacteria, yeast, nematode, fruit fly, rice and Arabidopsis, which is a plant. So the method used in DNA sequencing included two main approach. One approach is the expressed sequencing tag, which identify the gene expression as RNA, and another is the whole genome shotgun sequencing, means you are sequencing the entire genome including coding and non-coding region. Different steps of sequencing are: first the DNA were isolated, then they were broken into smaller fragments. These fragments were then cloned using bacterial artificial chromosome or yeast artificial chromosome. Then these cloned fragments were sequenced using automated DNA sequencer, which rely on the Sanger's method of sequencing. Overlapping fragments were arranged using computer programs. And finally, the chromosome mapping using polymorphism and microsatellite was done. Chromosome 1, which is the longest chromosome, was the last to get completed in the year 2006 because it is the longest chromosome. The main outcome or the feature of Human Genome Project, very important from exam point of view, that total DNA is 3164.7 million base pair. The average size of a gene is 300 base pair. Largest gene is dystrophin, which is 2.4 million base. Total gene is approximately 30,000, which was earlier thought to be 18 to in between 80,000 to 1,40,000. Further, it was identified that most of, that is 99.9% of DNA is identical in all the humans. 50% of the gene which were discovered, the function was unknown. Out of this, only 2% of the genome code for protein. A large portion consists of repeated sequence, which is important for chromosome structure and evolution. Chromosome 1, which is the longest, has most gene, and Y chromosome has the least gene because it is the smallest chromosome. 1.4 million single nucleotide polymorphism were also identified, which helps in study disease and human history through evolution. So these points you have to keep in mind, read it once, twice, question is directly asked from here. If we see the application, so it has several applications. It can help us to understand the DNA sequence, which will be helpful in biological research and medicine. It will provide, or it will give, a new approach to genetics in which we can study multiple genes at once instead of one at a time, because we have information about all the genes. This has the potential to impact medicine, agriculture, energy, environment, and forensic sciences. Now, this huge data need advanced computing tool, and it also need interdisciplinary expertise. The data which was obtained by Human Genome Project has further over the time lead to the development of DNA fingerprinting. So as we have seen in the Human Genome Project that 99.9% of human DNA sequence is identical. So only 0.1% variation is there, and only this 0.1% variation make every human unique. Now comparing full DNA sequence is very difficult. So DNA fingerprinting is a faster method for comparing the DNA sequence. This technique identifies difference in specific DNA regions, and this is called the repetitive DNA. DNA has a specific part called repetitive DNA. This repetitive DNA is small DNA sequence which is repeated many times. So DNA has two parts: bulk DNA which give a major peak, and satellite DNA which give a smaller peak. Now bulk DNA will have all the coding sequence. So bulk DNA is not used in the DNA fingerprinting. It is the satellite DNA which will be used in the DNA fingerprinting. Further, these satellite DNA will include the microsatellite and mini satellite. So overall, the entire DNA is divided into bulk DNA and satellite DNA. Further, satellite DNA is divided into mini satellite and microsatellite. This satellite DNA does not code for any protein, but it is useful for studying the genetic variation. Now, this satellite DNA will be used as DNA fingerprinting, which will be helpful for forensic science and paternity test. So, let's first understand what is DNA polymorphism. DNA polymorphism means the genetic variation in the DNA sequence. This polymorphism will be seen in the satellite DNA. So how this variation comes in these repeated sequence? So this is caused by mutation. Mutation in the coding or non-coding region. Now since we are talking about satellite DNA, so it will be specific for the non-coding region. Now if mutation is inherited and it is present in more than 1% of the population, then it is called DNA polymorphism. This DNA polymorphism is more common in non-coding DNA. Why? Because this non-coding DNA mutation do not show any effect on the individual. So if there is no effect, then the organism can easily survive because it do not compromise the reproductive ability of the organism. So such mutation are transmitted over the generation and it keep on accumulating over generation. So polymorphism accumulate over generation and it will cause a genetic diversity. So every family gene will have its own level of polymorphism which will accumulate over the generation and it will give rise to the specific DNA polymorphism. And this specific DNA polymorphism will be used in DNA fingerprinting, and it is a very important source of variation, so it is important for evolution and speciation also. This technique of DNA fingerprinting was discovered by Alec Jeffrey, and he used satellite DNA as the probe, and as we have just seen that this satellite DNA show high level of polymorphism. And these polymorphism or the satellite DNA, they are called variable number of tandem repeat. VNTR, they are the small DNA sequence is repeated several times and the size can be 0.1 to 20 kb, which differs between the individual. Different steps of DNA fingerprinting include: first, the DNA is isolated, that is, it is extracted from the sample of the body. Then it is cut into small part using restriction enzyme. In electrophoresis, it is separated by size using the gel electrophoresis. In Southern blotting, the DNA fragment is transferred onto the nylon or nitrocellulose membrane. After that, these DNA are hybridized using the radioactive VNTR probe, and finally it is detected by autoradiography. So let's understand this with an example. Every individual have pair of chromosome, one of it is coming from father and another is coming from mother. Now, each chromosome coming from father or mother have its own VNTR repeats, or it has its own polymorphism, and that define the nature of the individual, nature of the chromosome. All such chromosome together will form the DNA fingerprinting of the individual. Now here, as you can see, it is given chromosome number 7. So here chromosome number 7 has 2 copy because it is diploid organism, it is of human. So chromosome number 7, one of it is coming from father, another is coming from mother. So as you can see, the one coming from father has different tandem repeats and coming from mother has different tandem repeat. Similarly, chromosome number 2, one chromosome coming from father, another coming from mother, and both have different in tandem repeat because mother and father will have different polymorphism. Same with chromosome number 16, mother and father chromosome are having different in tandem repeat. Now, when this in tandem sequence are passed through gel electrophoresis, then they all will give different bands. Now, let's say there is a crime scene from where a sample is obtained. Let's consider sample C is obtained. Now there are two suspects, A and B. Now the in tandem repeats are run through gel electrophoresis. Now let's consider there are two suspects, A and B. Then gradually these two suspects also the DNA fingerprinting is done and then it is compared. So as you can see, if the individual is different, then the tandem repeat will not match. But if the individual is same, the tandem repeat will same. Now if you can see, compare C with A, we can see they are not matching. The in tandem repeat are different. Whereas when you see C and B, then here the in tandem repeat is correctly matching. So the DNA fingerprinting of C and DNA fingerprinting of B is matching. So who is the criminal? Individual B is the criminal, or it has DNA fingerprinting same as C. This DNA fingerprinting is used in forensic science, paternity testing, genetic research, population study, and wildlife conservation. DNA fingerprinting has wide variety of use. In conclusion, if we see the key feature of DNA fingerprinting, so every person has a unique DNA fingerprinting except identical twins. The VNTR, which is the small repeated sequence, show high polymorphism, and there is a unique DNA pattern for each individual. DNA fingerprinting uses a very small sample. Even a single cell is enough for getting the DNA fingerprinting. It is more advanced and precise than the traditional identification method, and it is widely used in forensic science, medicine, and genetics.
So I hope you must have find this video helpful. So if you like the video, please share with your friends who may get benefited from it. Also, comment me your queries if you have anything regarding this chapter. And if you want to cover any particular subtopic of this chapter in detail, then also let me know. So best of luck all of you for your NEET exam.